The title of my talk is The Bushes of Life Model and Human Ancestry in Light of Genetics Data. And before starting my talk, let me just advertise a book that came out last year. It's edited by myself and Samuel Lampa, a Swedish colleague of mine. We are both members of the Genesis, the Swedish creationist organization. And the first printing is sold out, but I hope the second printing will come out before the summer. And the topic, it's an edited volume, but parts of the contents is related to what I'm going to say today. And we have several well-known authors in the different chapters of this book. And then I also would like to invite you to Malmö in October this year, because then we have the first Nordic Creation Research Conference with several well-known international speakers, including Peter Borger, who is here today. So you're most welcome. And let me start giving an outline of my talk. So I will talk about the bushes of life model that God created animals and plants after their kinds in different groups. And I will, after having talked about that model in general, I will give a brief history of the systematic study of created kinds. It's also referred to as Baraminology. And then we will look at the three major goals of Baraminology. And the first goal is to identify these different groups or kinds. And this is ongoing work. And then related to that is also the second goal of the study of created kinds, to find the mechanisms of divergence and speciation within each kind. What is it that explains why we have some diversity within the different bushes or the different kinds? And then the third goal is to, once we have identified the different kinds and groups, to find the ancestral history of each kind. And then we will zoom in into humans and ask the questions, are humans a kind of its own? Are we all descendants of Adam and Eve who were created uniquely by God or not, without ancestry with other species? And then I will wrap up with some conclusions. So let me start talking about the bushes of life model. One could say generally that there are like three models for the diversity of life. The first one is a creationist species fixity model, whereby God created species ready, and then they haven't changed much over time. So it gives you sort of horizontal, vertical lines to describe graphically the history of life. The second model is the one I'm going to focus on, the bushes of life model, but it's also creationist, but it's rather that God created the first generation of each kind without ancestry between them. But then there have been divergence and speciation to some extent within all these bushes or kinds. And that's also creationist model. And then we have the evolutionary tree of life model, according to which all species descend from simple single cellular organisms. So if you look at the ancestry, that gives you a tree of life model instead. So you can say that the bushes of life model is between the species fixity and the tree of life models, because you have some common ancestry between closely related species, but not like as a large tree, but rather as local unconnected bushes. And let's first give some examples of tentative kinds and bushes. So I would say that humans, modern humans, different ethnic group or races, and historic humans, Neanderthals, Denisovans, Homo erectus, they are all descendants of Adam and Eve. And that would mean that humans form its own kind. Then we have like great apes, chimpanzees, gorillas, orangutans, they would form another kind. We have the dog kind of foxes, wolves, and dogs. they would form their own kind. And we have like cat animals, tigers, lions, jaguars, cats. They would also form their own kind. So these are some tentative examples of kinds. And before proceedings, could we just look at the Bible for a minute and see, is there a biblical motivation for kinds in general? And first we can start looking at the existence of bushes and kinds. And then we go to Genesis chapter 1, where God creates the whole universe and earth. And it says that God created plants after their kinds on day 3 and animals on days 5 and 6. And then humans also in the image of God on day 6. And then we proceed to chapter 2 in Genesis, where it's described in more detail how God created the first man and woman. So that's sort of the existence of God creating animals and plants after that kind. But what about the diversification within kinds? Could we find a biblical motivation for that? And I would say, in the Bible we read that animals and humans were commanded to multiply and fill the earth. And that gives us an interpretation that God, already from the start, he designed mechanisms for environmental adaptation, including speciation within kinds. That is, including formation of new species within kinds. And that is a completely different process than ordinary evolution. I would come back to that. And then we also have a limiting factor. When we look at the bushes of mammals and other animals, we have to take into account, from a creationist point of view, that there was a flood of Noah, which was an extreme bottleneck, where only 2 or 14 individuals of each kind, 8 humans, survived. As I mentioned early on when I gave the outline, the word Baraminology is also used for the bushes of life model. And Baramin, that's a Hebrew word for created kind. And it was Frank Marsh who in the 1940s introduced Baraminology as a word for the systematic study of created kinds. And as I mentioned when I gave the outline, there are 3 main goals of Baraminology. And it's also mentioned in the review paper by Leitner et al. First of all, to identify all created kinds, and that is ongoing work. Secondly, to find the mechanisms of divergence and speciation within each created kind. That explains why do we get these bushes from the first uniquely created generation. And then thirdly, once we have identified the kinds, find the ancestral history of each created kind. And this also relates to some extent to taxonomy. Taxonomic levels and bushes, because taxonomists, they sort life hierarchically into different levels. So starting from the bottom, we have species, we have genus, we have family, order, phyla, kingdom, and domain. So we have animals and plants. They are 2 kingdoms, the animal and plant kingdom. They belong to the eukaryotic domain of life. And then there are like 30 phyla within the animal kingdom and so on. And then we may ask the question now, a bush should typically correspond to the lower taxonomic level. So which is a typical taxonomic level of a bush of kind? Is it at the species, genus, family, or order level? And the creationist Erik Wassmann, more than 100 years ago, said that it depends on the study group, the taxonomic level. Whereas George McCree Price, he was also a creationist, he said that, well, yes, that might be the case, but family is often a good candidate. And I think these were very good predictions. So let us look at some possible kind levels. So as I said, I will argue at the end of this talk that humans form its own kind. So then a kind is defined, in this case, of the species level, including modern humans, Neanderthals, Denisovans, and are all descendants of Adam and Eve. But the other 3 tentative kinds that I mentioned, great apes, dogs, the dog kind and the cat kinds, they are taxonomically all defined on the family level. But there is recent research that indicate that chicken-like birds, galliforms, they are rather defined on the order level and could tentatively be its own kind. So that could be quite a lot of diversification within the kind. Let's now turn to a brief history of the bushes of life model and also these other models I mentioned about. And I would say that until the 18th century, most Christians were creationists and the species fixity model dominated. And moreover, the young earth creationism, that the world is very young, it was not only held widely among Christians, but it was mainstream in society. So the first edition of Encyclopedia Britannica around 1770 had several articles with a clear young earth view. And then the father of taxonomy, he was a Swede, Carl Linnaeus. He was clearly a creationist and he advocated species fixity, but late in life he actually changed his view to the bushes of life model. That's not so well known. Then we proceed to the 19th century after the work of the Scottish geologist James Hutton in the late 18th century and then Charles Lyell in the early 19th century. Most scientists adopted high ages based on the uniformian assumption that things change at the same rate all the time. And this was really important for tree of life models to emerge in the 19th century. We had the French scientist Lamarck, who had an evolutionary theory based on acquired characteristics in the early 19th century. And then Albert Wallace and Charles Darwin more or less at the same time published a theory of evolution where natural selection had a much more important role. But at the same time, there were several scholars who criticized the tree of life model, and they favored something that there were versions of this criticism, but it more resembled the bushes of life model. For instance, the British botanist William Herbert in the 1820s and the British biologist and paleontologist Richard Owen in the 1850s. But in spite of this, sort of these bushes of life views, when Darwin published his well-known Origin of Species, he equated creationism with species fixity. So that was for a long time the view that creationism was species fixity, although there were different points of view within the creationist camp. Then we proceed to the 20th century, start looking at the tree of life model, and even though Darwin's theory gained more and more acceptance, it had a breakthrough in the 1940s during the modern synthesis or neo-Darwinism, and that is explaining evolution by genetic mechanisms, and that is natural selection acting on random mutations. And we have some, for instance, the geologist George Gaylord Simpson, the British biologist Julian Huxley, the biologist Ernst Mayr, and the geneticist Theodosius Dobzhansky. They were all important in this process. And then it was highly believed that after the modern synthesis that the tree of life model was a solid scientific theory. But we could also look at what happened slightly before in the creationist camp. In the early 20th century, increasingly many creationists favored the bushes of life model over the species fixity model, including Eric Wassmann, that I quoted before, Harold Morton, George McCreary Price, Dudley Whitney, Harold Clark, and Byron Nelson. And then in the 1940s, as I mentioned before, Frank Marsh, he defined Baraminology as the systematic study of created kinds. And I would say that from the 1950s, species fixity became a minority position among creationists in favor of the bushes of life model, both for biblical and scientific reasons, I would say. So let's now return to baraminology and sort of this systematic study of created kinds. And as I said earlier, there are three main goals of this systematic study. Firstly, to identify all created kinds. Secondly, to find mechanisms of divergence and speciation within each created kind. And thirdly, to find the ancestral history of each created kind. So let's look at these three goals one by one. Start with the goal 1, to identify created kinds. And I would say that there are three main methods for identifying biblical kinds. And firstly, hybridization, observations, experiments. Secondly, the so-called cognitive method. And thirdly, statistical baraminology. Let's start with hybridization. And we will hear a talk on reproduction, I think tomorrow or in 2 days by Stefan. So then you will hear a talk by an expert on this. But suppose two organisms, individuals, can hybridize and have offspring. Well, that indicates that they must be closely related, because reproduction is an extremely complicated process from fertilization of the first egg and then to the and then during to embryological development with cells being differentiated in different ways. So they have to be related, these two organisms, in order to have viable offspring. And that was the basis for Frank Marsh in the 1940s. He suggested that hybridization is a sufficient condition for a pair to belong to the same created kind, because they cannot belong to different kinds, but species can still lose within the same kind. They can still lose the ability to have offspring. So it's only a sufficient condition. But in order for them to belong to the same species, more is required. Then they also have to have fertile offspring. But how should we define hybridization within kinds? Broadly, not necessarily only within species. Well, it's not required that the offspring are fertile. Like when we have offspring of horses and donkeys, we get mules that are fertile, that are not fertile, but they are still alive. But we don't even, the offspring need not even be alive. So in the early 1990s, the German Siegfried Scherer suggested that what is needed is that the offspring need to survive a certain stage of embryological development in order to count as a hybridization. And there is a lot of hybridization data from a book by Gray from early 1970s and the review article by Leitner in 2012. Let's look at one example from the cat family, which consists of 40 species and 14 genera. And hybridization data, here is an example from that book from the review article by Leitner, hybridization data of 10 species from this family that covers 6 genera, notated as G1 to G6. And then we see in this table, we have crossings indicate successful hybridization. And we see that we have, and this table might be incomplete, but still we have crossings over genera. And that suggests that Felida is a kind. So the bush should be above the genus level, according to this hybridization data. And then we have the second method, the cognitive method. And the idea is that it is possible for human senses to use human senses and sort species into groups, based on an article by Sanders and Wise from 2003. But each such group, it could be a very large group, like all mammals. It has certain common features and then includes several kinds. That group could also be part of a kind, like foxes. But even though the group that we recognize by human senses could be larger or smaller than a kind, it's still a useful tool in combination with other methods in order to identify kinds. And the biblical motivation for the cognitive method is that Adam was able to name all animals. We read that in Genesis chapter 2. That is, he was able to use his senses to identify kinds. And similarly, Noah was able to get land animals of each kind into the ark using his senses. So, and we also humans, we should also be able to use our senses to recognize different groups of animals. So here is an example using the cognitive method to identify the cat family. So we see a strong visual resemblance between the species of the cat family. And that, in combination with the hybridization data I show you, that sort of gives a cumulative argument for Felidae, the cat family, being a kind. So that was the second method to identify kinds. Let's go to the third, more modern method, statistical baraminology. And this is used for much larger morphological or molecular data sets, which cannot be analyzed by human senses alone. And statistical baraminology usually involves three major steps. First, you need to find a list of morphological or genetic characters, and for each species, list whether each of those characters is present or not. So here we see an example with 4 species and 10 characters, and the filled circles mean that those characters are present in that particular species. And then the second step is to compute the distance between each pair of species, like the fraction of characters that differs. And that gives us, in this case, in this specific example, a table with 4 rows and 4 columns, showing this distance between all pairs of species. For instance, the distance between species 1 and 2 is 0.1, because if we compare those 2 rows, we see that it's only 1 out of 10 characters that differ, character 6. It's present in species 1, absent in species 2. Whereas if we compare species 1 and 3, the distance is 0.9, because then it's the opposite. All but one of these 10 characters differ. It's only character 6 that is present in species 1 and 3. And then we fill out the whole table, and then we enter the third step of statistical baraminology, and that is to construct a heat map. We cluster the species into group so that distances that are small have a light color. Those pairs of species are given light color, whereas distances that are large between pairs of species correspond to dark boxes. And in this case, we have clustered the 4 species into 2 groups. Species 1 and 2, and 3 and 4, because there are small distances within each group, large distances between species of different groups. And let us give an example of a heat map based on real data. Here we have 34 animal groups or species. We have not 10, but 20,000 characters, which is molecular data, proteins. And I will return to this heat map later, but among these 34 groups, we have modern humans, we have and we have also some ancestral humans, and it turns out that they form their own group. So I will return to this later when I talk about human uniqueness. So let's now enter the second goal of baraminology. So how do we explain how these bushes form? Which are the mechanisms of divergence and speciation within each kind? And as I said, the biblical motivation for the bushes of life model is that God created each kind to rapidly adjust to changed environmental condition. And that includes 2 main features. First, God created diversity in the first generation of each kind. And then we also have designed mechanisms of change that act on this diversity from the first generation. And here we see an example of a kind with 4 species. At the top, we have created diversity in the first generation that God created. And then we see divergence guided by mechanisms of change that leads to 4 species corresponding to 3 speciation events. And I will return to this, but these design mechanisms of change are extremely effective, and that implies that the observed degree of speciation in each created kind would be possible, I would say, within a young Earth creationist timescale. But we also need to take into account other mechanisms of change. This is very important because there are also evolutionary, non-designed mechanisms of change, because we live in a fallen world after the fallen man. And they are much less efficient. They don't build up information. They erode. They are mostly deleterious changes corresponding to devolution, not evolution. But they act in parallel with these design mechanisms of change. So let's look into this in more detail. The created diversity in the first generations and the mechanism of change. And there are different types of created diversity. And this is a topic for a talk of its own. So I will not go into detail all these different types of created diversity of the first generation. Let me just mention a little bit about some of them. We have created heterozygosity. That has been known among creationists for a long time. Of course, the structure of the DNA molecule was not known until the 1950s, but something similar was sort of predicted a long time ago by creationists. And we know that if we look at the humankind, then it is founded by Adam and Eve. We as humans, we have 22 pairs of non-sex chromosomes. It could be the case that God created, Adam had 22 pairs of non-sex chromosomes, so had Eve. It could be that God created Adam's 2 copies of, let's say, chromosome 1 with small differences. So let's look at chromosome 1, for instance. So there could be some nucleotides where there were differences, and these differences are called single nucleotide polymorphisms. So, for instance, at one site, it could be the case that one of Adam's 2 copies had a C in that position along the coding strand, and the other homologous chromosome a T. That is one example. And that gives variation in the first generation that could lead to diversity. Then we had variation inducing genetic elements, and this was a name coined by Peter Borger, and I think we will hear more about this tomorrow, but this seems to be a very effective mechanisms of creative diversity. So it's like jumping genes and transposable elements. So here we see an example, a portion of DNA, the lilac DNA, is moved from donor DNA in dark blue to the recipient DNA, the light blue. And it seems that these mechanisms are very effective for quick changes later on. And I think you will hear more about this tomorrow. Then we have epigenetic structures that are above the genetic level, but they still lead to variability. And that is, for instance, they affect the way how genes are expressed. So here we see the DNA string wrapped around histone proteins, and the way these histone proteins act influences gene expression. But there are other types of epigenetic structures as well. Peter Borger also has another article from 2008, said that in the first generation there was also some redundancy, like in regulatory networks and so on, that gave some robustness and made it more possible to have environmental adjustments. And then we also have Randy Gyutza. He has introduced an engineering principle for environmental tracking. So it seems that within the cells we have input sensors, we have logical circuits and output actuators that make it possible for organisms to very quickly adjust to environmental changes. So these were some mechanism of variation in the first generation. Then we have all these mechanisms of change. And remember, we have 2 types of change, designed or non-designed. So we need to keep track of both of them in parallel. So population geneticists talk about genetic drift, when the different versions or alleles of a gene, they change sort of randomly over time. So it's and we also have founder effect when a new population founds. And they are typically regarded as non-design, but they can also be designed when the first generation, the founders, actually choose to change environment. In evolutionary theory, mutations are considered random copying errors, but there are many examples of also design mutations that are sort of increases the efficiency of adaptation to changed environment. And so one type of mutation breaks down, the other is efficient in terms of reacting to environmental changes. And then we have crossovers or recombination, that is reshuffling between paternal and maternal inheritance of DNA. And I would say that that's clearly design, God's way of increasing genetic variability. Natural selection, we know that that's a cornerstone of Darwin's theory of evolution, where sort of nature does the selection, but it could also be individuals doing the selection. So it could be of two types. Then we have the action of variation-inducing genetic elements, which can be very effective and change structures quite fast. We have epigenetic mechanisms that change gene expression. Genes can be switched on and off, and they are often reversible, so that if they can be switched on, they can later on be switched off. And then we will hear by Stefan a talk on reproduction, and that it connects to the embryo developing. And we know that parts of embryological development that, like Hox genes, they cannot mutate, but there are other parts of the developmental trajectory that seem to have some plasticity, as recent research indicates. And that could also be a mechanism for change within a created kind. So these were some of these mechanisms. Then we go to the third goal of Baraminology, to find the ancestral history of each created kind. So suppose a created kind has been identified, the first goal of Baraminology, and then we want to find the ancestral history of this kind. And that could be, we can ask about this history in different ways. We can look at the geographic history, and that includes biogeography, sort of geographic history in response to environmental changes. For creationist model often include that there was originally one continent, and then after the flood of Noah we had plate tectonics and continental drift. Some animals survived then through the debris rafting model. We have climate variations, for instance, ice age, a couple of 100 years after the flood of Noah. These are all parts of biogeography, how changing environmental conditions affect the ancestral history of each kind. But we could also look at this more genetically, and for each kind try to build its ancestral genetic tree. And then we need this mechanism of change I talked about in order to trace this ancestry. So, we can do this, for instance, from most mammals. They have Y chromosomes. So we can, and Y chromosomes, the males have Y chromosomes, and they pass it on to their male offspring. So we can use Y chromosome data to build male ancestral trees. So here we see an unrooted tree for human male data. And likewise, mitochondrial DNA is only inherited from the mother. So we can likewise build female ancestral trees using mitochondrial DNA data. And here we see an unrooted mitochondrial DNA tree for humans. And it's very interesting that these trees are consistent with a young Earth time scale for a wide range of species. And you can read more about this in publications by Nathaniel Jeanson and Jason Lyle. And look, you can do this for many kinds, many types of species, but let's now look at human Y chromosome and mitochondrial DNA trees. So we look at these two unrooted trees I mentioned about. So to the left, all tips are men alive today, and to the right, all the tips for the mitochondrial DNA tree are women alive today. And then we have some key questions. So we don't have the roots of these trees, so data only gives unrooted tree. So where should we put the root, the most recent common ancestor of all the tips of these two trees? And the second key question is, how fast do the branches coalesce, merge? And that is related to the question, when did the most recent common ancestor live? And that brings us to the question of the age of a tree's most recent common ancestor. And in order to understand that, we need to look at the concept of mutations, because here we see an example with an unrooted tree for 5 men, unrooted Y chromosome tree. And the branches of the trees, they differ because of mutation. When a father passes on a Y chromosome to his son, there are sometimes changes. And likewise for the female tree based on mitochondrial DNA. And the branches that differ by fewer mutations, they merge more quickly. And how quickly, that depends on the mutation rate and generation time. So here we see that, for instance, to the upper left, we have 2 Y chromosomes that when they merge, their common ancestor, along the path to 1 Y chromosome, there were 23 mutations and 28 mutations to the other Y chromosomes. But let's now assume that the root exists along this long branch with 86 mutations. So there are 25 mutations to the left of that hypothesized root and 61 mutations to the right of it. Then we can build a rooted Y chromosome tree where the most recent common ancestor is at the top and all the tips are at the bottom. And we still see the number of mutations along each branch. And now, in order to answer these key questions, we need to know the mutation rate, the number of mutations per generation. The faster the mutation rate is, the more recently the most recent common ancestor lived. And it's only in recent years that the mutation rate has been estimated with high accuracy. And using the so-called pedigree method, where for Y chromosome, you simply compare the Y chromosome of the father and the son and count the number of differences. You take the average of a number of father-son pairs and estimate the mutation rate. And likewise, you do for mitochondrial DNA data, comparing mitochondrial DNA between mothers and daughters. And this pedigree-based estimates of the mutation rates are much higher than previously believed. And that gives an age of the most recent common ancestor that is of the order 5,000 to 10,000 years for a wide variety of species. So that was the third goal of Baraminology, to find the ancestral history of each created kind or bush. Let's now zoom in and look at humans. And I will argue that humans are unique. It forms its own created kind, that we are all descendants of Adam and Eve that were created uniquely without common ancestry with other living beings. And I will do that in 3 ways. We will first use statistical baraminology to argue that humans are genetically different from great apes. Then we will look at non-sex chromosome data to argue that humans' genetic variation is consistent with ancestry from a first couple. And then thirdly, we will look at sex chromosome data, Y chromosome data, and mitochondrial DNA data to build male and female ancestral trees with a young Earth creationist consistent time frame. So let's look at the first of 3. Let's turn to the first case, using statistical baraminology applied to humans and great apes. And this is based on a paper by Leitner and Serati from 2019. And this is the heat map I mentioned about earlier. So now we are returning to this. It consists of a total of 34 species or groups of living beings. So we have 4 species from the cat family. We have 2 mice and rat animal species. From monkeys and apes, 18 species. Modern humans, and this is a group with modern humans, Neanderthals and Denisovans, they form the human group. And we have 4 chicken-like birds. We have 2 species from goose, ducks, and swan animals, and also an African frog. And this study is based on 20,000 proteins. And remember, in statistical baraminology, for each species or group, it's recorded whether each particular character, in this case a protein, is present or absent. And now these proteins can be located at different chromosomes for different species. So you need so-called sequence alignment methods to actually carry out this. And this is based on quite a lot of mathematical algorithms, such sequence alignment methods. But the result is extremely interesting. This heat map reveals that humans are clearly unique. Because here we see modern humans, and we also see Neanderthals and Denisovans. And then we also see the 4 species that make up great apes. The great apes, they are much more closely related, not only to monkeys, but also to mice and rat animals and cat animals than to humans. And this study is based on an enormous amount of data. So in my view, this study alone more or less proves human uniqueness. Let's now turn to the second method of sort of arguing for that humans are unique. And that is looking at non-sex chromosome data to support human uniqueness. And then we will look at human DNA variation. I mentioned a little bit about this earlier, that as humans, we have 22 non-sex chromosomes, 22 pair of them, and their total length is 3 billion nucleotides. So here we see all these 22 non-sex chromosomes listed, and we have, as I mentioned earlier, there is some variation between humans, and a biallelic SNP is a position with 2 possible nucleotides. So an allele is a version of a nucleotide. And as I mentioned, for instance, we could have along chromosome 1. It could be that in some humans there is a C allele or a C nucleotide at that position, whereas for others there is a T nucleotide. And for each such SNP, we can define something called the minor allele frequency, and that is the frequency of the less common allele. So assume, for instance, that for this particular nucleotide that this picture illustrates, 60% of all men have a C in that position, and 40% of all human copies of that position have a T. Then 40% is a minor allele frequency because it's less than 60%. So the minor allele frequency is always between 0 and 50%. And then we have of the order 10 million biallelic SNPs along our genome, human genome. And that's a huge number, but it's still only 0.3% of the whole genome. So we are mostly genetically identical. So there are very small, few positions where we differ. But now one can build something called the allele frequency spectrum. And that is simply a histogram of the minor allele frequency of all these 10 million SNPs. And the interesting thing is that the shape of this histogram gives some information about the history of humanity. So for a creationist model, you would say that an allele with a large minor allele, I could just say that for a young population would tend to have a flatter allele frequency spectrum than an old population. So for a young population, there would be more SNPs with a large minor allele frequency, closer to a 50% distribution between the 2 alleles of that SNP. And for a creationist model, then the interpretation of this histogram would be that an allele with a large minor allele frequency to the right of the histogram typically corresponds to created heterozygosity that I mentioned about earlier, God putting variation in Adam and Eve along their 22 pairs of non-sex chromosomes, whereas an allele with a small minor allele frequency to the left of the histogram, that would, on the other hand, pinpoint a more recent mutation. Whereas a tree of life model in evolutionary theory, you don't have a concept like created diversity. You only have mutations in neo-Darwinism. So then a large minor allele frequency to the right will correspond to an old mutation, and a minor allele frequency that is small would correspond to a more recent mutation. And I actually gave a talk on this 6 years ago at the ELF. In 2018, and the year after that, I published a paper with Anne Gauger where we fitted human allele frequency data to a unique origin creationist model, and we had quite a good fit, and the same did Sanford et al. In 2018. And this does not prove human uniqueness, but it still shows that it is possible, or a creationist model explains genetic variations of non-sex chromosomes as well as the evolutionary model. So let's now move to the third case of arguing for human uniqueness. Now we will look at female and male ancestral trees. Let's start with the female trees, mitochondrial DNA human tree, arguing that it's consistent with a young Earth creationist timescale. And then we go back to the 1980s, and then the first estimates of the age of the most recent common ancestor were like 100,000 or 200,000 years. And she was referred to as mitochondrial Eve. You might have heard about that. But these estimates were based on evolutionary assumptions, like comparing mitochondrial DNA between humans and chimps, and then assuming we had a divergence 6 million years ago, and then comparing mitochondrial DNA among humans, and that gives an estimate of like 150,000 years ago that we had the mitochondrial Eve, or the most recent common ancestor of all females. However, in the mid-1990s, the first estimates of the mutation rate by this much more reliable method that does not rely on evolutionary assumptions from the start, but is more based on like empirical data, the pedigree method, they gave conflicting results, but generally the age of the most recent common ancestor was found to be at least 10 times as small as the previous estimates. And then in Nathaniel Jensen, he actually pooled all these studies in order to get larger data set, and he obtained a most recent common ancestor less than 10,000 years ago, consistent with when Eve lived according to a literal interpretation of Genesis. And he also identified 3 major branches emerging from the root that could potentially be the 3 daughters-in-law of Noah. Then we look at the history of how our knowledge about the human Y chromosome tree emerged. And there are some similarities with this history, but some differences. Because for Y chromosomes, you cannot use the phylogenetic method because the Y chromosomes of chimps are very different from the Y chromosomes of humans. But so the first estimates of the most recent common ancestors of, and I would say that is by itself an argument for human uniqueness, looking at the Y chromosomes of chimps and humans. But for this reason, in the 1990s, the first estimates of the age of the most recent common ancestor were also gave ages of several 100,000 years. But this was based on low quality DNA data. And then later came studies based on higher quality DNA data. And Nathaniel Jensen and his colleague Holland, they pooled several of these studies with high quality DNA data and obtained an estimate of the mutation rate with the pedigree method I mentioned about, which was much higher, leading to a most recent common ancestor that lived 4,005 years ago. And that is consistent with Noah, because according to the Bible, all men are descendants from Noah. That's the most recent common ancestor, not Adam. And this is quite remarkable. And in 2022, Nathaniel Jensen published Traced, a book where he makes further use of this Y chromosome tree, not only to identify Noah 4,500 years ago, but he also finds 3 branches emerging from the root that would correspond to the 3 sons of Noah, and then branches further down along the tree he identifies with descendants of Noah a few generations later, mentioned in chapter 10. And then he also sort of manages to explain a number of well-known historical events like foundation of civilizations, and also the American African slave trade in the 70s and 80s centuries and so on from this tree. And he says himself that there is some lot of work to be done. Some suggestions are tentative, but it's really strengthened the case a lot for the creationist model. So that completed my look at humans and human uniqueness. So let me end with some summary and conclusions. So we have been looking at Baraminology, the study of created kinds. And as I mentioned, it involves 3 major goals. First of all, to find all created kinds. Secondly, to find the mechanisms of divergence and speciation within each kind. And thirdly, to find the ancestral history of each kind that corresponds to an ancestral tree. And this is an ongoing field of research. So I mean, these kinds, all kinds are not identified yet, but new data is emerging, so there is lots of open access molecular data available, genome-wide data, ancient DNA data, and there are many tools at our disposal, like statistical baraminology, I mentioned, sequence alignment, tree building algorithms, and so on. And the results indicate that data show clear grouping into kinds, but still a lot of work remains. Secondly, that designed mechanisms allow for very quick adaptation within kinds. And this is something very different from evolutionary theory, where evolutionary theory is thought of as building up new information. We will hear this tomorrow from Peter Borger's talk, that the information was already there in the first generation within each kind as created the diversity. And thirdly, analysis of mitochondrial and Y chromosome DNA indicates recent history of each kind in agreement with the young Earth creationist timescale. And then we looked at humans specifically, and I argued that genetic analysis confirm the Bible's view of human uniqueness and human history. And my prediction is that future research based on more data, we have large data sets being built up, for instance, the Earth's Biogenome Project, will continue to strengthen the Bushes of Life model and human uniqueness. And because human uniqueness is so central in the Bible, and we heard God created us humans in the image of God, and that is also explained in the first 2 chapters of Genesis. So I hope that also this kind of research will make people to realize how reliable the Bible is, how valuable humans are, and that will lead to that they give their life to Jesus. So let me end with a quote from the Bible, from the Romans chapter 1, verses 19 and 20. What may be known about God is plain to them, because God has made it plain to them. For since the creation of the world, God's invisible qualities, his internal power and divine nature, have clearly been seen, being understood from what has been made, so that people are without excuse. And that is what he has made. That also includes genetics and DNA and things like that.